I think we'll get started. So hi everyone, thanks for joining us today for the introduction of FreeSurfer. And it's my pleasure to have Ran and Dev here to give us the presentation. So Dev will give us the introduction first, and Ran will take over and give you some examples on how to use it. So without further ado, go ahead, take it over.
All right, thank you so much for organizing this and putting it all together. My name is Dev, and this is Ren, and we both work for the Laboratory for Computational Neuroimaging, otherwise known as the FreeSurfer Lab. Some people call it the Bruce Fischel Lab, whatever it is, we work for it. And so our lab basically developed the software FreeSurfer that can take MRI images and do all kinds of fun analyses on it. So I'll be giving an overview of that. And so, can you guys see my next slide okay? Awesome.
Yep.
Okay, so basically what we'll talk about is I'll kind of do an introduction of what FreeSurfer is and how it works, and then also how to use it at the Martinos Center. And then Ren will kind of go into troubleshooting and what to do when FreeSurfer fails, because unfortunately it is not a perfect software package. So sometimes there's manual intervention kind of necessary. And so she'll kind of go over that, and then also where to go for help when maybe you can't figure out what to do or you can't figure out what's wrong. So FreeSurfer has been cited over thousands of times about various different topics. And so, it's not specific to if you're only studying Alzheimer's, even though that's a lot of the research that our lab does. It's been used for all sorts of topics. And there's a lot of publications done using FreeSurfer, which is pretty exciting. It's really gained popularity over the last decade. And so we provide a bunch of citation info on our website as well if you're looking to cite and use FreeSurfer in your projects or your papers. And so FreeSurfer, we're really used to, in this day and age, we're used to our beautiful interfaces, right? And you get to click on, if you're using Safari or a Mac, you get to click on like the download button, and then you get a little notification and there it is. And you kind of work with softwares that way. Whereas FreeSurfer operates out of a terminal, which I think we typically associate with hacking or code or I don't know, bank robberies. At least that's how I interpreted it when I first came to the lab. But really what this does is it is a developmental tool that allows a lot of flexibility within FreeSurfer. It's why we're allowed to add so many different components to it, over just the course of it operating. We're continually getting to update it, and it also provides you flexibility as you're processing your subjects, because it can allow you to add additional arguments to how you want your subject processed, or it allows you to batch process subjects, and it kind of allows that consistency across your subjects, and then that makes for a better experiment, more robust. And so there's a lot of benefits that come with FreeSurfer operating out of a terminal, but it's definitely, at least for me, was a little bit intimidating. And so we provide a lot of resources to kind of give you a little bit more comfortability with the terminal and with using Linux. And so that's available on Mac, obviously Linux computers, and Windows. And it's available to you at the Martinos Center, and actually on any computer. And so what FreeSurfer does is it will take an MRI image that you've acquired, typically an MP-RAGE, T1 weighted image, and it will then create this 3D reconstructed volume that's been labeled, segmented, parcellated, analyzed, and it'll give you a bunch of other volumes as well as statistics based on that one input MRI image. And so how FreeSurfer works, kind of how you get it started, is you run this command in this red box here. So this is kind of the main part of FreeSurfer, right? So you run recon all, and that's kind of how we're going to talk about it for the rest of this talk. And recon all basically means to reconstruct that MRI image that you gave it initially, and it will go through all of these steps and provide you with statistics and different volumes about your subject. And so what you see here is the pipeline of recon all. This is basically what FreeSurfer does. So you give it your T1 weighted image, usually a DICOM or a NIfTY file. It'll do some skull stripping, which is self-explanatory of it takes out that surrounding dura and skull, and then it will go through and label the brain. It'll do some intensity normalization to kind of differentiate gray and white matter, and then it will segment white matter on its own, and then it'll kind of inflate, it'll blow up the brain, which is a very crass way of putting it, but it will inflate your brain and compare it to multiple other atlases. And this provides some robust segmentation. And then it'll generate surfaces based off of that inflation. And then it will additionally label those surfaces. And then from there, it'll generate statistics. So a lot gets done in this one recon all command, and I'll kind of break down what this command is and how to use it. But this is what gets FreeSurfer started. And so a little bit more info on the input image. So it does need to be T1 weighted image. Actually, that's a lie. I think you can specify other volumes as well. But typically, and what it has been used long-term for, is T1 weighted images. And what that means is that the white matter is brighter than the gray matter. So it's actually true to their anatomical names. And FreeSurfer is used to using full brains. It is not necessarily best for subjects under the age of 5, just because it's using these atlases and it's comparing against a bunch of adult brains. And so if you throw a pediatric brain in there, it's gonna FreeSurfer will get confused, and it won't be able to kind of match up the gyri or the sulci to the adult brain the way it would with a infant brain. And so that's why we suggest using this with adult input images. Although we are working on, one of our PIs is working on, infant FreeSurfer as well. And that's just because their brains are changing at such a rapid pace. And so, like I said, it's just comparing to adult atlases. So you won't get quite the accuracy of results that you would with an adult brain. We also don't use FreeSurfer on non-human primates. And so if you're studying gorillas, we're sorry. But we can do humans. And so it can handle some pathology, but if they've had a major TBI or stroke, that can cause FreeSurfer to fail in some ways. And Ren will kind of go over some different ways to combat that. And so with FreeSurfer, so you are in your terminal, you run your recon all command, and then you get this 3D volume. And it's important to recognize that it is a 3D volume and to know what that means. And so that means you're going to get the brain in 3 different views, right? Our sagittal, axial, and coronal. But then as you're going through the brain, you're going to look at it as a 2D image. And so you're going to look at it in one of these planes, whichever you'd like to use. And you're looking at different voxels, and FreeSurfer has gone in and characterized each of these voxels and kind of separated them into different surfaces. And so these voxels correlate with the resolution of your scans. And so the higher the resolution, the smaller the voxels. And so if you think of, if you've taken a picture and it's really pixelated, that means it was a low resolution picture, because the voxels are very large, and so you're not getting a lot of that definition. And so what FreeSurfer does is, based on these atlases, it's going to go through and it's going to segment your brain based on the intensity of each of those voxels. And so an important differentiation that you guys should be aware of is that segmentation, when we talk about segmentation, is often referring to those subcortical structures. So the tiny little things beneath the cortex. And then when we talk about parcellation, it's talking about the actual labeling of the gyri of the brain. And so we use 2 different atlases to do this, and you can specify which one you'd like to use. But so there's the Desikan-Kiliani atlas, I may be pronouncing those wrong. And then there's the Destro atlas, and these are our collaborators that have worked on this from the very beginning. And if you want info on how these atlases were constructed, if you're doing specific analysis on that, or for your papers and citation purposes, that's on our website as well. But yeah, so that's going to be a little bit important later on in Ren's talk, so she'll probably touch on this again. But this is one of the, or I guess technically two aspects that FreeSurfer provides, is the segmentation of subcortical structures and parcellation of the cortex itself. And so, and I think we just want to really emphasize that these are separate analyses. So you get your labels, your segmentation / parcellations, there's a lot of jargon, and then you get your surfaces. And so I kind of like to think of it as, these are the super colorful things that FreeSurfer will provide you with. And these are the outlines that you'll get. And what these outlines kind of demonstrate is the white matter surface, the yellow you see here, and the peel surface, the cortex, which is the red line that you see here. And so when FreeSurfer kind of inflates, you get vertices, and that's what gives us our 3D structure, 3D reconstruction. And so again, it's really amazing that FreeSurfer does provide a lot of different volumes for us to look at. So if we keep a running count of what I've talked about so far, we have, the original T1 image that we gave it. And so we have just a nice T1 weighted image. We have the segmentation of subcortical structures. We have the parcellation of the cortex. We have these surfaces. And so you can see this one is the white matter. And then if you think about building on top of that, that's what gives you this peel surface, or like the cortex. And so this kind of gives us insight into like the sulci of the brain, whereas this gives us the more kind of what we think of when we think of a brain. And then it also generates heat maps that you can view your data in or do analysis in. But the important thing to know is that FreeSurfer it cares about the white matter the most in terms of editing purposes, because once the white matter is created, if we want to go back to this step, it kind of segments out that white matter, and then it will kind of grow the peel surface from there. And so going back to this slide, it kind of starts here, and then it will generate the peel surface based on that white matter surface. And that's going to be an important point when it comes to troubleshooting, or when you're trying to fix a bad recon, is making sure that you're making appropriate edits where they need to be edited, so that FreeSurfer can regenerate those areas as applicable. And so, like I said, recon all refers to reconstruction. So we're reconstructing that T1 weighted image that you gave it, and creating all of those many different volumes that I just went over. And so this is, like I said, your main command line that you're going to give FreeSurfer to kind of get it all started. It's the catalyst of FreeSurfer analysis. And so obviously you're going to want to give it a input volume. You want to tell it what you want it to process. So this can be a DICOM file or a NIfTY file, and you'll want to specify, obviously, with the appropriate extensions. And then you can also run this DICOM unpack command, and so that will give you a list of the different acquisitions that you are inputting into FreeSurfer. And so you also want to give your subject a name. I was telling Ren, I'm not sure if FreeSurfer was created at a time when our bosses were having children. I don't know if they were spending a lot of time with Elmo, but a lot of our examples are Elmo related, with Bert and Ernie here. And so, for instance, he named this subject Bert, and that's kind of what you'll see in a lot of our examples. If you attend any more of our tutorials, it's always Bert. And so you want to name this subject that, because it might not be in the DICOM file. Some of our DICOM files are very numerical or just a little bit vague. And so you want to name the subject, and whatever you name this subject is what FreeSurfer will create a subject directory for it. And then it'll create a bunch of other files beneath that. And so this is important because you'll have your main subjects directory of your analysis. So if you're studying Alzheimer's patients and normal controls, you will have, once you specify the subject name, and you specify where your subject directory is, FreeSurfer will then create a subject directory within where you specified it. And so you just want to make sure that you're giving distinct names, because like I said, like if you have Jenny in both Alzheimer and control, you want to make sure that you're not overwriting anything, and that each subject has a unique name, whether that's unique to its pathology or however your naming conventions work with your studies. It's just important to give it a unique name. And then this dash all seems a little bit redundant, and it will make more sense when Ren goes into her presentation, but basically it's telling FreeSurfer do everything, because technically you can run FreeSurfer for only certain portions that you want. Say if you're editing a recon and you only need to redo that one section, because as you saw before, it's a very long pipeline with a lot of different steps. And so when you're initially running a subject, you're going to want to do a dash all flag so that FreeSurfer knows to run it through the entire pipeline. And this used to take quite a few hours, 10 to 20 hours to run a couple of subjects, but luckily this time, as they have come out with different versions of FreeSurfer, we've cut that down, and it can run anywhere from 4 to 8 hours now, which is a lot better than 10 to 20. So that's the command line for FreeSurfer. And now we're going to go over a little bit of what gets created in these subject directories. So every time you run recon all, you'll get a new subject directory. So for instance, this subject's name is Bert. So when FreeSurfer has completed running through the entire recon all process, these are the main directories you'll get: scripts, MRI, surf, label, and stats. I can guess that you guys can guess what's in each of these directories, but I'm going to go over them anyway. And so the BERT directory in total is only about 400 megabytes. So it's important to keep that in mind when you're reconstructing your subjects, and you want to make sure that you have enough room on your computer to, see all of these subjects to completion. And so it's just important to keep that in mind. So your scripts directory is going to have probably the most important document in there is going to be the recon-all.log. And basically, this is going to tell you every command that FreeSurfer ran in while it was doing recon all. This is really important when your subject finishes. You're going to want to go in and check and make sure that it ends at the very bottom with subject finished with no errors. And Ren will talk about that a bit more, but this is going to be really important for once your subjects are finished going through the pipeline. The MRI directory is where all of those volumes are stored that we kind of went over before. And so in the recon all process, FreeSurfer generates a lot of different files, and each step in the pipeline will produce different volumes. And so all of those volumes, wherever they are in the process, are saved in MRI. And so the most important ones that you'll probably want to look at once your subject has finished is your orig or your T1, your brain mask, your white matter mask, WM.MGZ, and your ASEG. And so these two are ones that you're going to be making edits to if something went wrong. And your ASEG has all of those subcortical segmentations and cortical parcellations. Try saying that 5 times fast. And the difference between these is that the raw average is kind of very similar to your DICOM, your original input, in that it's in its native anatomical space, whereas all these other volumes that FreeSurfer has created, it's kind of put it into a registered anatomical space to all of those other volumes. And then this is where your surfaces are. So like I said, FreeSurfer gives you a lot of different volumes. And so it, nicely enough, will separate those for you. So MRI is strictly the kind of, they're 3D volumes, but they are, you can easily recognize them as an MRI of a brain. And then surfaces are kind of that more 3D volume that you can kind of turn around and make all kinds of fun presentations out of and stuff like that. And so this is where you can kind of see how the brain has been inflated and what those surfaces look like. And then in your label file, this is where those labeled gyri that were compared to those atlases that I went over, this is where these ones live. And it kind of labels each gyri for you. And these are important to check if you are particularly interested in, maybe there's a certain strip of cortex you're interested in or Brodmann's area. These are important to check to make sure that FreeSurfer did a good job of labeling them accurately. And then lastly is the stats directory. And so this is where FreeSurfer not only made pretty colored maps of the brain, but it also computed a lot of analyses on the volume of different subcortical structures, the thickness, the what else? Intracranial volume of the brain. It just generates all these really useful statistics that you'll likely use in your studies and prints them in different text files under your stats directory. And like I said, back to the different atlases that you can use, it will also generate and specify kind of which stats are referring to which atlases. So that can be useful if you're interested in that. And so this is what those stats files kind of look like. And it gives you a nice table of contents, and then it will list out everything you want to know about this brain. And there's a couple of commands, aseg stats to table and aparc stats to table, where you can kind of specify which structures you're interested in. And so, for instance, I was recently studying the hippocampus, and I was only interested in hippocampal volume, but you have all of these different structures, the white matter, the cortex, the ventricle, which are great, but I don't need them. And so these commands are super helpful for specifying just the structures you're interested in and still getting all of that super useful information. And there's a lot of, if you just run back to kind of FreeSurfer operates out of a terminal, you can just run the command you're looking for and then --help. And this will print out in your terminal, it will print out the instructions for using that command and the different flags you can use to specify what information you want, what structure you want from these statistics directories. So those are super helpful when you're getting to kind of that analysis part of your study. And so to do all of this, you have to download FreeSurfer. And so we have a wiki page that kind of gives you step-by-step instructions for Mac, Linux, Windows, and other operating systems. And so those are super helpful. So if you're trying to, go it alone, definitely check out our wikis that will help you download FreeSurfer onto your machine. That way it'll be easy for you to use locally. And so, like I said, FreeSurfer is available to us at the Martinos Center. And this is where it kind of gets tricky. If you're not super familiar with how to use a terminal, you're going to learn, and it will get better. I can say, as someone who did not use a terminal ever before I started this job, and now 2 years in, I'm super comfortable in it. And it didn't take me 2 years, which is exciting. And so basically, the way that I like to think about it is you have to source FreeSurfer every time you use it. And so I like to think about it in terms of if you have a sticky note on your refrigerator, and it kind of has all your reminders for the day. And so before you leave for whatever you're up to for that day, you see the sticky note and it kind of reminds you like, oh, okay, I have to go do that. That's kind of what this is like, in that you have to kind of remind your computer, hey, this is where FreeSurfer is located, and this is the version I want you to use. Go for it. And so it's really important to source FreeSurfer using these different commands. So this is because in the install instructions, you have to download your FreeSurfer app into somewhere on your computer. And so this is basically, you're spelling out for your computer where FreeSurfer is located on it. And then there's also a dev version. So right now we have version 7.1.1, and it's kind of the stable release that we have. And then you can also source the dev version, which is kind of funny because it was really tricky when I first started this job because I felt like everyone was saying dev all the time, but they weren't referring to me. They were referring to the developing version of FreeSurfer, which is pretty cool because that means you have front row access to all the new tools and tricks and shortcuts and things that our development team is adding to FreeSurfer and Freeview. So I definitely recommend it. It's really interesting and neat to see the new things that they're constantly coming up with. You can also have a bash RC. And so this is kind of like a more permanent version of a sticky note in that it will automatically source FreeSurfer for you every time that you get ready to, every time you open up a terminal. And so basically you would put that same command that you would use if you were just sourcing it in your terminal, and you would put it in this file and then add stable 7.1.1 or dev at the end of your bash RC. And so if you need help kind of setting this up, we're happy to help. We also have a lot of examples on our website, and so feel free to use those. But this definitely makes life easier if it's just sourced automatically and you don't really have to think about it. And so lastly, the other resource available to you at the Martinos Center is you can use Launchpad, which is I'm gonna butcher the actual name of it, but it is a large, it's like a powerhouse of a computer essentially. And so you would just give it this pb submit command, and then your username, and then the recon all command that you would use. And it will it's useful in that it's going to process it on Launchpad and not on your local computer. So it's not going to take up that memory or power on your own computer, but instead it kind of gets submitted as a ticket to Launchpad. And then kind of going back to the statistics, you can also generate your own statistics in that you can kind of label your own regions on the brain, and then you can generate stats from that. And so basically, you can use this command MRI surf to surf or MRI vol to vol. And there's a lot of documentation on this, but basically you can generate these things based on whatever you're studying. It doesn't have to be specific to the subset of structures that FreeSurfer offers, but you can kind of create your own, or you can use alternative atlases to compare your labels to. And so how to generate these new stats is you can manually draw them on yourself. So right here, you're seeing the hippocampal subfields, thalamic nuclei, and the brain stem. And then you can kind of generate stats based off of these drawings, even though that's a very basic word for it. It takes a lot more attention to detail than that. But this is useful if you're studying just particular regions of the brain that FreeSurfer may not have covered in the ASEG or APARC. So that is kind of a very quick overview of FreeSurfer, how it works, and what you can do with it. And now I'm going to kind of pass it off to Ren, who's going to chat about, it's kind of a doom and gloom thing going on, but what happens when FreeSurfer messes up and how to fix it.
Thank you, Dev. So for the second part of this presentation, I'm going to go over some of the common errors that you may find after you have run Recon All. So although like FreeSurfer is a very powerful software, it still makes some mistakes. Dev, can you change the slide, please? Okay, so essentially, we can categorize the types of failures or errors that you may come across into two categories. So you may often see as a hard failure or a soft failure. And the hard failure essentially is when Recon All quits before completing running through the whole pipeline, as Dev described. And so A soft failure is when recon all finishes, but the output needs some type of modification. So you will have to go into your data and make certain adjustments so that your data is accurate. Thank you. So where to locate your hard failures? So as Dev described, FreeSurfer creates a specific subjects directory for your data. And so it creates 5 additional directories. And in your scripts directory, you're going to look for a log that's named reconall.log. And so if you open it up, you'll actually see a message that says recon all exited with errors. So that is the indication that this is a hard failure. And so some of the causes for hard failures is perhaps you may have some permission issues. You may need to get permissions to access, certain files. You may have run out of disk space or RAM, as well as perhaps your sample could have certain pathological conditions, such as your brain might have low contrast or some abnormality that FreeSurfer isn't able to identify. So what to do if you come across a hard failure? So as I said, check your reconall.log error message, then go through your data and assess how your sample is, if there's good contrast. You could also rerun the step that failed, verify the output from the last successful step, and perhaps a common step that some of our users do is they could search the error type in our mailing list. So if you search for keyword of recon all with error, you will probably see a response from a previous lab member that tackled on this question and helped another user solve it. As well, you can also run the modified version of your command if needed, or you can send us an email with your reconall.log file. So when you do send us an email, please provide the reconall.log, as I mentioned so many times, but as well as your operating system and hardware, as well as your exact command line that you try to run. And this will just help facilitate communication between our lab members and with your team. All right, so now moving on to soft failures. So as I described, soft failures are a bit tricky because if you open up your reconall.log, you will see that reconall has completed without errors, but that doesn't mean that your data is perfect. So I'm going to go through some of the errors that you will find that are soft failures. So these to the left, you can see that we have some skull stripping errors, white matter or automatic segmentation errors, some intensity normalization errors, peel surface, and topological defects. And I'll explain what those are. But essentially, to fix the errors, you cannot fix your surfaces. So as Deb mentioned, FreeSurfer labels different surfaces for your data. However, you actually need to go into the volumes and change the volumes and then reach that and then go through recon all, and that will regenerate your surfaces. So a few things to keep in mind when you are analyzing your data. You have to make sure if your surfaces follow the gray matter or the white matter borders, as well as does your subcortical segmentation follow intensity boundaries. And the cool thing about FreeSurfer is you can actually go back and correct these errors. Some morphometry packages don't allow this. So it's also a really good learning tool and a better way to just have better data for your studies. So these are the types of manual interventions that you most likely will perform on your volumes. The first is erasing voxels, filling voxels in, cloning voxels, and adding control points. Now, when you do make these edits, you should only take about 30 minutes or less. Of course, the first time you do it's going to take longer, but with practice, it shouldn't take 3 hours. And if you do feel like you're running into an editing problem where you're spending hours and hours on your data, then perhaps it's better to send it over to us and we can, quality assess it. But essentially, after your manual interventions, you can rerun parts of Recon All. So this is kind of, I talked about that you need to edit your volumes to make your corrections. So the volumes that you will most likely edit are going to be your brain mask and your white matter volumes. And so those can be found in your MRI directory. And this beautiful chart on the right is kind of a cheat sheet to help you to kind of guide you as to what volume you should edit. So for example, if you want to erase voxels, you should edit your brain mask. If you want to clone voxels, you should edit your brain mask as well, or add control points. But for your white matter volume, you want to make sure that you're only erasing voxels or filling voxels in. So Recon All, Dev did mention that Recon All essentially, it can be described in parts, and essentially it is made up of these different auto recon flags. So we have auto recon 1 and auto recon 2, auto recon 3. And so these flags essentially create the entire processing pipeline. So for example, here in the sculpt stripping, you have auto recon 2 and an intensity normalization as well as white matter. And once you reach surface extraction, that's when auto recon 3 runs. Now this is really helpful for you because perhaps you only need to make changes to your white matter segmentation. And all you have to do is really just run the command that's given there, recon all, with the auto recon 2 flag. If you don't have any other changes in the skull stripping or the intensity normalization. So that actually saves a lot of time on your part. And if you want to learn more about these different parts of Recon All, you can definitely check out our FreeSurfer wiki. Right now we have our 6th version table up. We do have a current version of FreeSurfer 7.1.1, but it's in the working process. But essentially here in this table, you can learn what type of input you need, the command line, how it works, the type of flags that you will use. So it's a really good learning guide. Or you can just make your life easier, which is what I do as well. It's just running Recon all together. And now, since we're kind of transitioning a bit into identifying and the certain errors, certain soft failures, and applying certain edits to fix them, we have this really nice tutorial on our wiki where you can actually make changes to example data or to your own data. So this tutorial will actually walk you step by step on what to do. So for the first soft failure, this is probably not so common, but this is in the category of skull stripping failures. So in this case, as you can see, when FreeSurfer processed your T1 image to your brain mask image, it removed too much. So at this step, FreeSurfer, you just want FreeSurfer to remove the skull, but unfortunately, it actually took out part of the cerebellum, and you don't want that. So there's a few things that you can do. You can either use a clone tool, and that's on Freeview, or you can adjust the watershed parameter. And what do I mean by the watershed parameter? So it will be included in your new recon all command, but essentially this watershed parameter tells FreeSurfer how much to remove from your volume. So a higher number means to strip less. And so we provide the number 25 to start as kind of the baseline, but you can play around with this number. And the good thing about this command is that you can run it and it takes usually 2 minutes to run. So it's very quick and not so tedious. So this is another example of a skull stripping failure, but it's kind of the opposite from what we saw before, where not enough was removed. So as kind of Dev talked about it, so for the surfaces, FreeSurfer tries to identify and detect where the boundaries are for the white matter and the peel surface. Now, if you look in the picture, the peel surface actually extended to include a blood vessel. And we don't want that because that's inaccurate anatomy. So you may see sometimes that FreeSurfer may include the dura or the blood vessel as part of your peel surface, which is not okay. So the quickest way to edit this is to just open up your brain mask volume and erase your voxels that are aligned to or that correlate to the blood vessel or the dura. So essentially just eliminating the anatomical skull structure that you don't need, and then just rerunning this command. So this is another example, similar in the same category, and this is pretty common, where you will see that the eye socket is actually being misclassified as white matter, and we don't want this. So easiest way to fix this is to erase some voxels erase the voxels that essentially represent the eye socket in your white matter volume, and then just rerun the command below. So although this image is interesting because although it appears to be a little funky where you see that the skull is included, it actually does not really, it doesn't affect your surfaces. So you can actually leave it alone because what we care about mostly is the surfaces, and we care about our cortex, not necessarily the skull. So moving forward to segmentation errors, examples of this are when white matter is misclassified as non-white matter and vice versa, and gray matter could be classified as white matter as well. So some of the causes for this are intensity normalization failures or partial voluming. So for the first segmentation error, this actually occurs, I believe, in older cases, so in older brain samples. But you will see that there's these hypointensities where here the gray matter is all misclassified, and in the white matter you see like a little voxel of gray matter, which is kind of not true if we think of it anatomically. So what you just need to do is essentially fill in your voxels in your white matter volume and then just rerun this command below. So this is kind of a good example to analyze, because if you have the sample and you look at it just in your coronal view, you will kind of question the labeling in the region where Dev is highlighting right now. So this misclassification of the gray matter kind of looks a bit abnormal, but if you actually look at it in your sagittal and axial views, it's actually just a sulcus that is being represented, and that's a good recommendation when you use FreeSurfer, is to always think of your brain as a 3D volume. But of course, analyzing 2D, just remember to reference that back. So moving forward, this is another type of error that you may see in some of your data, and it is intensity normalization. So in the left picture, that is the uncorrected one. And so you can see that FreeSurfer kind of failed to include parts of the white matter. And so we want to make sure that FreeSurfer properly includes all the pieces of the white matter. And what essentially you do is you just add control points to your white matter volume. And by adding controlling points, you're just telling FreeSurfer to essentially include these points the next time it runs through recon all. So it's kind of just like help guiding FreeSurfer to make sure to identify other regions with similar intensities that could also be white matter. So this is another error, and these are more likely, analyzed in a 3D sense. So these are topological effects, and they can occur in all parts of your brain, but mostly they are categorized into two sectors, so holes and handles. And to fix this, you just really need to go into your white matter volume. But essentially, the causes what is a hole. So a hole occurs most likely due to partial voluming, where white matter or gray matter looks like non-white matter, and it is segmented as non-white matter and creates a hole. And a handle essentially is when there's perhaps some types of interference that's bright, has a similar intensity to white matter, and it is misclassifying it as white matter. So as I mentioned before, all you really need to do is edit your white matter volume. And if you do have holes in your data, you just really need to fill voxels in. And then if you have handles, then you just need to erase voxels. So this is kind of as well as an analysis tool that you can use. In FreeSurfer, we have, as Deb mentioned, we have the option of adding a heat map or representing our data using a heat map. And so essentially, if you look at it through the 3D volume, you see that where the hole is, or where the hole is being described, and you reference back to your 2D volume, that is not a hole, that is just a misclassification of white matter. And so what you can do, and this gives you a good tool to analyze your images in the 2D view. And all you really have to do is just add the voxels in to get rid of this hole. So essentially, when you make those changes, you will see that your surfaces are way cleaner, way smoother. Yeah. Okay, next slide. So this is kind of a good point to mention, is that if you look at your subcortical region, you will kind of notice that it is a little funky looking, but essentially we're not gonna, when you are analyzing your surfaces, you don't really need to worry too much about the subcortical region. And maybe if you click on the next slide as well. As well as for ASEG. So ASEG does a separate analysis that is distinct from our surfaces. And so if you look at it here, kind of the blue ribbon, or it looks a bit funkier, it looks like it's not anatomically correct. But because we are looking at the surfaces, we don't need to worry about the subcortical region as well. So as a summary, kind of breaking down into parts, if your peel surface includes too much, then make sure to edit your brain mask volume. However, if it affects the white surface where too little is included or too much, or if your peel surface includes too little, then go ahead and edit your white matter volume. If this is a segmentation error, of course, or add control points if this is a normalization error. So these are kind of just some extra bullets to remember when you are quality assessing your data. So for hard failures, check your reconall.log file. That is a pretty clear indication whether or not you have a hard failure. For soft errors, your surfaces are the ones that are being affected. But although your surfaces are affected, don't edit your surfaces. Instead, edit your volumes and then run the specific recon all commands, and then that FreeSurfer will regenerate those surfaces. You can also use different types of editing modalities, such as erasing voxels, filling voxels in, cloning voxels, or adding control points. And the volumes that you will most likely use are your brain mask and your white matter. You also have the option of using brain.finalsurface.mgz, but that one is not so common. I think that's for special purposes. If Dev, you want to add on to that.
Yeah, I think, sorry, I couldn't find the unmute button. Yeah, I think it used to be a lot more utilized in editing, but for now, I think Ren said it best, and that we're just using our brain mask and our white matter mask.
Thanks, Deb. And going into detail or just giving a general summary of your different types of soft errors, of course, for skull stripping, watch out if too much is removed or not enough is removed for your segmentation. Keep an eye out for misclassifications of white matter or gray matter. And just kind of a side note, FreeSurfer does keep track of your edits. So that's really nice for future purposes. And make sure when you are analyzing your data to look at your multiple views, not just your coronal view or sagittal view. And if you don't know whether or not to make an edit, just leave it alone, or send us an email and we'll help you make those changes. And last but not least, your edits should remain less than 30 minutes, perhaps even 15 when you become an expert. And so just to reiterate, definitely check out our wiki. It is a great tool to learn how to use FreeSurfer and how to make edits and how to use FreeSurfer as a tool for your studies. If you have any questions, of course, check out our email archive, search up the keywords that are related to your case, or you can also type in the flags recon all and then -- help to kind of know what FreeSurfer is doing in your terminal. And as well, last but not least, send us an email and we'll be gladly to help you.
Thank you so much for the awesome talk. It's very informative. Oh, there's not an announcement. Sorry, I thought it's over.
No, it's okay. It's okay. Deb pretty much covered it. Essentially, we have a newer version. It's FreeSurfer 7.1.1. It has more upgrades. It's super fast compared to 20 hours. And SamSeg also has lots of improvements. So definitely feel free to check it out.
Okay, now we're done.
Now we're done.
Yes, and there are a few questions, and thank you, Deb, for answering the question on the fly. And there's still a couple more in the Q&A box, and some people ask questions in the chat box too. So can you guys help answer the questions?
Yeah, so this question about if you have high field structural data, I believe there's a 7T flag that's literally just -7T that you can use that can kind of let FreeSurfer know to analyze that data differently according to the field strength. And then I know we are coming out, if we don't already have a hippocampal subfield protocol, Jean Augustinak did a lot of work with that. She's our neuroanatomist. She kind of is in her own lab now. And there might be a paper on it. So if you look up FreeSurfer hippocampal subfields, I think they've published something on it that might help you out with that. What is the recommended command line to resample and resize non-MRI files? So for example, resampling the WM Park or ASEG to match the dimension of T1. MRI vol-to-vol might be a good one. What came to mind for me was MRI register. And so again, our wiki, if you just, one thing that we like to mention is when you're searching in our wiki, use the text button. I know this is so minute, but use the text button. It'll show up more results than if you, type in your search items and then hit titles. But I would suggest going there and looking up MRI register, because I think that's the one that you're going to want to use to resample and resize. To run them in batches, I think that goes back to the it goes back to the Launchpad cluster that I talked about. I'm trying to find the slide, but basically this is a really helpful way of you just submit multiple subjects. So you, I know we have scripts available, but also I cannot find the slide for the life of me. Basically, you can either submit multiple command lines to Launchpad, and then you can have, 17 subjects running. And that can be super useful for, found it 10 years later. And so this is super useful in terms of it will give you notifications of if it ran to completion, if it exited, and it'll kind of send it right to your email, which is why you input your username there. And so basically you can just submit multiple of these, and that's how you can batch recon quickly. And then we also, oh, sorry.
Put the comments here. So Paul just from IT at Martinos just gave a talk 2 weeks ago about the new cluster we're switching to. So it's called the MLSC cluster. So if you missed that seminar, please go to the Martinos YouTube channel to check that talk out, because they will be using a new cluster for batch computing.
Yes, they will have far more intelligent things to say about it than what I just tried to explain. If you wanted to do group stats, we also have a longitudinal pipeline, which I don't know if we mentioned, which is really helpful because basically what it does is it will process all of your subjects through recon all, and then it will create a base template and then compare each of your time points to that base template, and it will generate longitudinal stats for you. So definitely check that out if you're interested. And then group stats would be something as simple as using the ASEG stats to table command, and that will allow you to pull statistics from multiple of your subjects directories. Whew. Okay, I really wanted to get a quick explanation of longitudinal pipeline, but we could have a whole talk on that. But I think that's all the questions.
There are 2 questions in the chat box. So one of those is there anything in the pipeline to segment head CTs?
Head CTs, I believe so. I know that FreeSurfer takes structural data, diffusion data, PET, fMRI. I'm not super sure about CT, so I can definitely get back to you. But my inclination is that it does, and that it would label the head CT the way that it would with other volumes, using ASEG or the APARC or whatever you're interested in.
Okay, I think that's all the questions we have. Thank you so much for the talk.
Thank you.
Thank you for having us.